You're listening to the Good Question Podcast with Richard Jacobs. Our goal is to make each of our guests exclaim, that's a good question. I don't know the answer. Because when that happens, it means you, the listener, may be inspired to learn more beyond the interview and to ask great questions yourself that lead to new insights. In this podcast, we cover historical and current anthropology, comparative religion, and history. Welcome, and let's get started.
Hello, this is Richard Jacobs with the Good Question Podcast. My guest today is César de la Fuente, or César de la Fuente. We're going to talk about getting new antibiotics from looking at the DNA of Neanderthals, woolly mammoths, animal venom, and now proteins of ancient microbes. Very, very interesting stuff. César is a professor at the University of Pennsylvania. He runs a group called the Machine Biology Group. He's one of the youngest tenured professors in the history of Penn Medicine. So, very interesting. Welcome, César. Thanks for coming.
Thank you, Richard. Great to be here.
Yeah, tell me about the Machine Biology Group. What is that?
Yeah, so that's basically, it's the group that I direct at University of Pennsylvania, and it's this really transdisciplinary team that encompasses people with a wide range of expertise, right? So, for example, we have experts in computer science, experts in chemistry, experts in microbiology, engineering, even physics, all working together in this really incredible environment. And I'm really lucky to be able to work with people that come from all around the world, come to my lab, to this group, to try to push the boundaries of science and the boundaries of knowledge. So it's really a fun, I'm really lucky and fortunate to be able to work with such incredible people.
So how did you get the idea? And well, first of all, why antibiotics? Why is it important that we pursue new ones? I don't know if people are aware of what's going on with current antibiotics.
Yeah, there's a huge global health crisis, so that we call antimicrobial resistance, or the acronym that we use is AMR. And these are bacterial infections that are increasingly, resistant to every drug that we have available. And those drugs, we call them antibiotics. So these infections are currently associated with about 5 million deaths per year around the globe. And if we don't do anything about it, if we don't come up with no solutions by 2050, that number is projected to double to 10 million deaths per year, becoming the number one cause of death in our society, even surpassing cancer. And so, we take that very seriously. I think it's one of the greatest existential threats to humanity. And so in my team, we're trying to think outside of the box to see how we can come up with new molecules that might be able to target bacteria, and also how we might be able to find new molecules in places where perhaps people haven't looked before, or how do we develop tools, computational tools that can enable us to accelerate the pace of discovery.
So why look in the DNA of like ancient animals, or in these far off places? They wouldn't be exposed normally to the I would think, to the bacteria that that cause us problems. I mean, why not use organoids, and why not try to deal with the bacteria that are killing people, but in a lab environment or in a mouse model or things like that?
Yeah, we also do all of the above, but so essentially a lot of the work that we have done, you can cluster it in two main areas. One, we're trying to mine the code of life for new functions, for new antimicrobial functions, for example. So we've developed methods that enable us to mine or explore what nature has produced, right? We think of evolution as this planetary scale optimization process that we can learn a lot from it, and we can extract a lot of useful molecules and useful information from it. So we've done that, and we've done that by mining extant biology, but also ancestral biology, like you mentioned, to try to find new antimicrobial molecules. We've been able to successfully find functional molecules in the human proteome, which is encoded in our genetic code, but also in our closest ancestors, Neanderthals, Denisovans. And then we scale that up to all of ancient biology. And so we took this journey with our AI systems through the genetic data of organisms like ancient penguins that were extinct in the 50s, or magnolia trees that disappeared throughout evolution, or even woolly mammoths, ancient elephants, giant sloths, and many, many other creatures that used to roam around our planet at one point. But unfortunately, through extinction events, they disappeared. But now we have this opportunity where we have access to some of their genetic data. And the way we think about this is we think of molecules as documents of evolutionary history, where we can extract, potential therapeutics, but also learn more fundamentally about how evolution did things and how and perhaps why evolution did certain things. So that's one whole cluster. And the other whole cluster is that in addition to learning from Mother Nature, we're also developing generative AI models that can create new to nature, completely artificial molecules, and that belong to the realm of the artificial, right? They're molecules that evolution has never thought of, but we think perhaps they give us a fighting chance at targeting antimicrobial resistance and perhaps other major problems facing humanity.
Yeah, I guess there's a lot of ways to slice it. What about, I don't know, I know they wanted to make antimicrobial materials, maybe materials of compounds that bacteria would never normally see, that have like a high roughness factor, so that, like bacterial membranes would get shredded, kind of, getting close to them. I don't know, I guess there's all different angles, but...
Yeah, I mean, if you think about it, right, some of the contemporary pathogens that we're exposed to today, right, in the world, most likely they've never seen some of these molecules that existed, many, many millions of years ago. So on the therapeutic side, you can think of this as a way of extracting potential therapies that can help us address present day problems, like the problem of AMR, by looking at the past, by using this ancient data essentially as relics or fossils that we can bring back, right? We can synthesize in the lab, and we can, in some cases, some of these molecules that we've identified, they are no longer, as far as we know, expressed in nature today. So they really kind of, in some cases, disappeared throughout evolution. And so we're bringing back those molecules to in this case, to counter drug-resistant infections.
Yeah, but why would they be any more likely to fight an infection versus, I don't know, like, the bark from a tree along the side of the road that grows, I don't know. It just seems odd. Like, why would that be a better area to look at than anything we have today around?
Yeah, so we've also done that. We've also explored extant biological data. So we're doing all of it, right? So we don't think it's necessarily better or worse. We just think it helps us expand the combinatorial sequence space that we explore in evolution, and people had not really done it before at this scale. Looking at, both extant and ancient biology as a source of antimicrobial molecules. So we think, from a fundamental perspective or a curiosity-driven perspective, we think it's interesting because we can learn about how evolution did certain things and how, certain mutations, incurred modifications in the function of those molecules.
So I understand you want to expand the combinatorial space. That makes total sense. What's, even if you do develop a new antibiotic, though, I mean, I don't know, from what I've heard is Big Pharma is really the only ones that have enough money to get it through clinical trials, and they don't want it because it's like a 10-day dose and you're done. So who's are there government grants for this stuff, or how do you get this stuff to market even if you find it?
Yeah, you're absolutely right. It's extremely difficult. It's really hard to fundraise to actually develop molecules through IND enabling studies and through clinical trials. So we've spun out a company in an attempt to try to do that, but we know it's going to be really tough, like you mentioned. But nevertheless, the way we think about this is that ultimately it would be for the benefit of humanity, right? We were discussing earlier how these infections, they're a big problem, right? They're associated with 5 million deaths per year in the world. And I think also importantly, and perhaps this has not been highlighted sufficiently, is that without working antibiotics, modern medicine as we know know it, it would really collapse, right? Because you need them for a lot of routine interventions in hospitals, things like surgeries, even like a C-section, organ transplantation. You need them for childbirth, and you need them to treat immunosuppressed patients, for example, cancer patients. And so really, they are a cornerstone of a lot of the medical procedures that happen every single day in hospitals around the world. And so I think it is important to think of trying to develop new antibiotics as a sort of like a benefit to humanity as opposed to trying to make profit, even though, of course...
Are there any that have been made that are just sitting there with lack of funding?
Yeah, so there are instances of antibiotics that have been approved, but then hospitals do not have the incentive to use them right away. They only use them as a last resort situation. And so it's hard for companies to kind of to obtain a profit, right? So some of the ideas that people have thrown around is perhaps getting government subsidies involved, or nonprofits that perhaps could do something about this and help fund some of this stuff to help ensure that companies are are successful ultimately, right? And to be able to incentivize other companies joining this area. The other thing that maybe you find interesting is that we're also experiencing a decrease in the talent pool because there is less and less funding to do this kind of work in AMR. We're getting less PhD students trained, and there's sort of like a brain drain that we have experienced over the last decade or so, and so that's also concerning, right? Because we need to train the next generation to help us combat AMR.
What about phages? I mean, a bacteria, they have to still have phages that kill them. Otherwise, they would spread all over the place. How come no one looks at that? Or at the protein coat of various bacteria that are AMR to see what's changed? Like, why not a more direct focus?
Yeah, that's a great idea. There are a number of groups around the world doing very incredible work in phages. So just for your audience, bacteriophages, these are viruses that only infect bacteria. So they don't really do anything against human cells. And people have attempted to leverage phages as a way of treating drug-resistant infections. And in fact, if you study their history, they have this really interesting history where they were using the Soviet Union. Even today, if you go to the Republic of Georgia, you can buy them at pharmacies in little cocktails, to treat infections. And so I think that's a really valid alternative to conventional antibiotics. I think the more ways and the more different approaches that we try to develop to counter AMR, the better for the world. So I think those are all great avenues to pursue.
Are there commonalities if you look at the exterior membrane of AMR bacteria?
So in some cases, the membrane can change, or bacteria that become resistant, they can do that in a number of ways. One of them is by changing their membrane. You can think of it as they change it to sort of shield themselves more effectively from the outside world and from antimicrobials. But they can also degrade antimicrobials. They have these efflux pumps that enable them to pump out antimicrobial molecules. So they have a huge arsenal of mechanisms that enable them to survive, essentially, right? Let's not forget that bacteria are some of the most ancient organisms to have ever existed on Earth. And so they have a huge toolkit of little tricks that enable them to essentially survive and adapt, right? And so, yeah, I'm no longer surprised by the incredible, the clever tricks that they have up their sleeve to kind of overcome any situation.
And what else? They use efflux pumps. I mean, what else can they use? What are some of the things you've seen?
Yeah, they have enzymes that enable them to. So when they come across an antibiotic molecule, they can degrade it or break it down. They also form biofilms. That's an adaptive response where they form these multicellular communities of bacteria. So they form these little houses, these little sort of towns and cities. And what they do is they encapsulate themselves in this, what is called a matrix, which you can think of it as a really effective shield all around the whole microbial city, and that matrix is composed of things like sugar and DNA and proteins. And so conventional antibiotics cannot really penetrate through that really thick and effective shield. And so that's another way that they can do it. They we can talk for a lot longer, but they just have a wide range of survival strategies, essentially.
There's certain bacterial genus that tend to form these defenses more effectively than others. Gram-negative, gram-positive.
Gram-negative bacteria. So you can classify bacteria, like you mentioned, in gram-negative and gram-positive. Gram-positive bacteria only have one membrane, one thick membrane, and gram-negative bacteria have two membranes. They have the outer membrane, and then they have the inner or cytoplasmic membrane. So the bacteria that are most problematic in hospitals, that kill the most people, are gram-negative pathogens. And if you look over the list from the World Health Organization, the usual suspects that you'll find there as the pathogens most dangerous to our society, those are primarily gram-negative. And that's because they have two membranes, so it's really hard to kind of penetrate into them with molecules. And so they include names like Pseudomonas aeruginosa or Acinetobacter baumannii, almost unpronounceable names, but that are essentially these really clever pathogens that can cause a lot of harm in humans.
Are there any bacteria that are really good at competing or outcompeting other bacteria, regardless of whether they become antibiotic resistant or not?
Absolutely, yeah. Bacteria. So essentially, in any environment, bacteria, the way they survive is by colonizing a particular environment, and then in the process of doing that, and in the process of growing their colonies, they have to outcompete surrounding microbes, and so they're really experts. A lot of them are really experts at doing that. For example, we've studied a bit in detail Pseudomonas aeruginosa, which is a gram-negative pathogen that is highly effective at outcompeting surrounding microbes. But there are many, many others.
Are there any ones that are harmless to people, even if temporarily we have a whole ton of them in us, but they're very good at outcompeting, bacteria we don't want?
Yeah, that's absolutely true. So there are the vast majority of microbes that exist in the world are actually beneficial. They're not pathogenic. It just happens that the pathogenic ones, because they kill humans, they get a bad reputation. But in Anonas, we have millions of microbes that live in our mouth, on our skin, in our urinary tract, in our intestine, that do a lot of good things for us. Actually, they have co-evolved in many cases with us, and they help us digest our food. They help us fight off disease. They outcompete pathogens, and so they do all these really good things for us. And a lot of them are really, like you mentioned, really experts at fighting off actually pathogenic bacteria that would be bad for us, for the host.
Again, is there any commonality in how they outcompete? I don't know. Are there so what happens with these antimicrobial resistant bacteria? You throw all the antibiotics at them. Are they just unfazed by them? Are they hurt by them but not as much? Are they using the antibiotics as somehow raw material to even grow faster and stronger? What kind of dynamics do you see?
So yeah, it's a combination of all of the above, and it really depends on the microbe. So in some cases, they overexpress these efflux pumps that enable them to pump out, the toxic antimicrobials that we throw at them that are toxic for them, right? In some cases, they form biofilms, so the antibiotics can no longer penetrate into and reach those microbes to kill them. In some cases, they produce little molecules that operate like a form of chemical warfare to kind of kill surrounding microbes. And so there's all these different tricks, right? And the way that one of the ways that resistance can spread in a microbial population is in plasmids. These are circular pieces of DNA that can be easily transmitted between microbes, and that they confer resistance, through different mechanisms. But is this really cool way that microbes have of sharing genetic information in these circular DNA structures to kind of ensure the survival of the whole community, of the whole microbial community.
Any promising compounds that you found that you can talk about? The mechanism of action, whatever you can say.
Yeah, absolutely. So in our whole efforts to mine the code of life, so that means genomes, proteomes, metagenomes with machine learning models. We've been able to identify, for example, mammoths 2, which is a molecule encoded in the woolly mammoth proteome that is quite effective even in mouse models of infection at reducing infections. We found elephasin 2 from the ancient elephant, and really a lot more. So in summary, a lot of the work that we've done has enabled us to discover millions of potential sequences with antimicrobial properties by really mining biology at scale. And out of all those, we have probably by now synthesized thousands of them using chemical methods. So we have these chemical robots that enable us to synthesize whatever we design or discover on the computer. And then we've probably tested and validated in preclinical animal models, hundreds of them. So really, we're doing this at a pretty large scale.
What do you think will be possible in the next year or so? Have the AI models got stronger to where you compare many more millions of compounds now? Like, what do you think is going to happen?
So what's already happened is that the AI models are getting really good, really reliable, really accurate at predicting antimicrobial function. And this is because we're training them with really high quality, standardized experimental data. And this is a hypothesis that we proposed a number of years ago. We call it the standardized dataset hypothesis, where we predicted that if we trained our models with really good quality data, their performance would scale with the amount of data that we fed them. By performance, I mean simply the accuracy of those models. And so when we started to track these metrics, I think it was in 2021, the hit rate was around 60%. And by hit rate, I mean when we actually make those molecules through synthesis in the lab, how many are actually active against bacteria in ground truth experiments. Well, that accuracy right now is at 95+%. And that's because our models have gotten increasingly accurate and reliable. And, so we've basically almost solved that problem, obviously in a very specialized, narrow area or for a very specialized objective function, which was to predict whether a particular molecule will be antimicrobial or not. But now that we've reached 95+% accuracy, I think we can, we're doing a lot less experiments than we used to, right? And I think that's quite telling of what some of these AI tools can do, that if you train them really, really well with high quality data, they can become incredibly good at really specialized fields, right? And so what we're doing now is we're extrapolating that, and we're creating high quality data sets for many, many other parameters, including toxicity, stability, folding, PK/PD profiles, all these other parameters that you actually need to make a drug. And we're trying to train models in each of those areas to then have them achieve 95+% accuracy. And in parallel, we're also working on AI agents that will be able to tap into each of those models as needed to give a really high accurate response to the user.
Where can people find out more about your work and follow up? What's a good centralized place?
Probably they can check out our website. We also post on social media, so we can share that information on this podcast.
Okay, very good. Well, César, thank you so much for coming on the podcast. I appreciate it.
Yeah, thank you so much for having me.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
Thank you for listening to the Good Question Podcast. Please email support@thegoodquestionpodcast.com if you have any referrals to great guests for us to interview. Visit thegoodquestionpodcast.com to hear more interviews. And please help us spread the word by rating and reviewing us on Apple Podcasts, iTunes, Spotify, YouTube, or wherever you listen to this podcast.